Thank you very much for the invitation, Mike, to this technical beekeeping group of the ECBA, Essex County Beekeepers Association. My name is Craig Story, and I teach at Gordon College in Wenham. I've been teaching, this is my 16th year teaching, and in my teaching duties I teach a basic cell biology class for freshmen, and a lot of them haven't had biology in a while, and I'm assuming that a lot of you haven't had biology in a while. So I'm not going to assume any background knowledge, so to speak. So I will try to start the basics and work our way up to some things that maybe have practical applications in your own hives. But yes, thanks for the invitation. And I find that the best way to learn a topic is to teach it. So that's why I volunteered to teach on honeybee genetics. I just moved. I hope the camera's pointed in the right direction. That's good. All right, so actually Gordon has a beekeeping club. They named it like this club, GCBA, Gordon College Beekeeping Association. So it sounds very, very fancy, but it's really the bee club. And we now have an observation hive in the Kennelson Science Center right across from my office, which has been going for about 3 weeks, and that's been fun to do as well. So I've been a beekeeper since 2015, and I'm like all of us, we're learning all the time. And I knew a few things about bee genetics, some curiosities about their sex determination. We're going to talk quite a bit about that in the first part of the talk. We're going to talk about different ways that sex is determined in the animal kingdom. I will also talk about some of the developmental pathways that lead. Whoops, what is going on here? There we go. So basically the first half of the talk is on sex determination and some practical aspects of how that was discovered originally and what we know about it today. I will end on something very cutting edge, RNA interference, but there's only one slide on that because it's pretty complicated. And I will also talk about different genetic traits that are found in bees that are of interest. Now, when you think of genetics, right? You think of a single person that's known with regard to genetics. And I should have worn my t-shirt. It has a picture of this guy. That's him. He's the one on the upper right looking at the flower. See that? He's not even looking at the camera. It's Gregor Mendel, right? And so we speak of Mendelian genetics. That's referring to Mendel. When did he live? Well, he lived and did his work in the 1800s, 1860, around in there. And you may know that's around the same time that Langstroth did all of his stuff, 1850s. That's when beekeeping really got started. And I learned through preparation for this talk, I learned that Mendel actually tried doing his genetics on honeybees. But it's not as easy as peas. All right, peas are a lot easier. Yeah, and so we'll see why, as the talk goes on, we'll see why that is. Okay, so we think of our genetic complement as like a deck of cards because we get a hand delivered to us when our parents produce eggs or sperm. One produces the eggs, one produces the sperm. I think you know this, right? Those packets contain genes that are blended together from the parent, right? And it's a random process. So at the time of Mendel, it seemed like you have these two plants, say, or these two organisms, they get together, and then they are a blend of their parents. So they had this idea that the genes blend. Mendel came up with the observation that that is not true. That's not the case. They may seem to blend, but then they separate out again. So they're actually what he would call particles. They're like entities that don't blend. They don't mix. All right? So this is the particulate hypothesis, the idea that parents pass on discrete heritable and heritable units that encode our features, our traits. And so do you see how that's a very different idea than the blending idea? Now we know now what those are, what those genes are, all right? And Mendel did things like he took some true breeding purple flower pea plants and some true breeding white flower pea plants that, in other words, they always produced white flowers, the other plant always produced purple flowers, cross them together, and you would think, oh, if it's the blending, you'd get like a light purple or something. But no, you got all purple, right? And then he did the next thing. He took all of those purple first generation plants. We would know those if you're a farmer. You'd say, oh, those are hybrids, right? and then mated all those to each other, and voila, you got the white back again. See, there's no blending going on. There's this particulate thing. And furthermore, it was in a 3-to-1 ratio. All right? Wow. Now, we know now that the purple is encoded in a gene that helps produce pigment, the purple pigment. And if you have one copy of the pigment, it gives you a purple flower. If you have 2 copies, you get a purple flower. Right? The only way to get a white flower is to have no copies of the good gene that makes the purple pigment. And so if you take those parents' genes and you make a little grid, you can see there's a 3-to-1. There's one that's purple-purple, and 2 that are purple-white, and 1 that's white-white. Do you remember doing this in high school, maybe, perhaps? All right, so that explains the 3-to-1 ratio. And then he looked at a whole bunch of other, you're thinking to yourself, I thought this was a bee talk. No, we'll get to the bees in a minute. So he looked at a whole bunch of different traits, not just the purple color, but the constricted pod, yellow pods, tall versus short. He found a whole bunch of the 3-to-1 ratios happening. All right. A couple of genetics terms to keep in mind that will probably crop up as I go along. When people speak of things like the purple versus the white, that is a term we call the phenotype, right, of the individual, whether it be a plant or a bee or a fly. It's the appearance, the phenotype. If we refer to the purple gene or the white gene or the non-purple version of the gene, we call that the genotype. So do you see that, like I said, there's 2 capital P, there's 2 in each of the offspring. If it's capital P, we say it's homozygous, meaning that the 2 versions of the gene are the same. If you have a capital P, small p, that's heterozygous. The 2 genes are different. You still get the same phenotype, which is purple, right? See, I'm using those terms to get you familiar with it. Little p, homozygous. And then another term, recessive, okay? A recessive gene is one that gives you a lack of something often, all right? So Mendel's experiments were done around 1860, but it wasn't until the 1950s that people figured out that the gene was made of DNA. But even before, well before Watson and Crick discovered the structure of DNA and figured out how it replicates by complementary base pairing and all that, nucleotide stuff. You know, you may have heard of this. This happened. It took a long time, but way before that, people had this idea that the chromosomes had something to do with heredity. And the chromosomes are visible by light microscopy. They're pretty big. Yeah. So the chromosome tends to look something like this. And here we see another term that's coming up for a couple of terms. So you'll notice along these chromosomes, there's a long arm and a short arm. These particular chromosomes happen to be duplicated. In other words, the cell is about to divide, and those 2 components of each are going to be separated from each other. So it's a duplicated DNA. But you'll see there's the locus, all right, the position along the chromosome where that gene for purple flowers is located. You can have 2 versions of the gene, and this is the term allele, okay? An allele is a version of a particular gene, right? It can be a functional allele that gives you the purple pigment, right? In that enzymatic pathway to make the pigment, make the flower purple, or it can be a non-producing, a non-functional allele. And so some genes, including some that we're going to talk about in honeybees, have many alleles, many varieties in the population. So we all know that we want a lot of genetic diversity in our beehives. In other words, we want lots of various alleles present there. Not all, like if you keep mating the offspring back to the parent, eventually it's called inbreeding. You have less diversity. That can lead to more recessive genes coming out like this. This is a human example where you see someone who has a lack of a pigment-making gene that causes the hair and the iris of the eye to be non-pigmented. That would be albinism. All right, and this diagram is nice. It follows upon what we saw before, which is you can see the sperm and the eggs on each of the sides of the little rectangle, little square there. And then you just fill in the blocks according to a mathematical kind of fashion, and it gives you the ratios expected in the offspring. It's called a Punnett square. I know some of you are shuddering, like, oh, Punnett square. Oh, I remember that. Forgetting that. All right, so it's actually not that bad. It's just a way of distributing the alleles mathematically and getting the probabilities expected. Yeah. So we will talk about a recessive gene for honeybees a little bit later. Okay, so let's switch gears slightly now and talk about sex. All right, so we're going to talk about the chromosomal basis of sex. Now, in humans, when we speak of the term sex in a biological sense, we're talking about the physical characteristics that typically go along with one or the other sex in male versus female, typically. And of course, there are variations. There's variations among this, even the human population, but for the majority, there is a typical male, typical female, and that's governed by X and Y chromosomes, as you may remember, right? So in mammals, we have X and Y chromosomes as a special pair out of our, how many pairs? 23 pairs, right? So it's 22XY for a male and 22XX for a female. The Y chromosome is just pathetic. Look at this. Look at this little pathetic thing, right? See that? Compared to the X, it's much, much smaller. It is pretty pathetic, but it's powerful because it can make a male, all right? So what's on the Y chromosome is one single gene that initiates a pathway to maleness, right? If you don't have that, you're going to have a female pattern of development. And so these still pair with each other and so on because they have the ability to do that properly. But in terms of sex determination, it's the XY system. All right. So now who decides, in terms of the parents, whether the offspring is going to be a male or a female? In this case, in humans, it's, who is it? The male. Because the female has two X's and only makes X eggs, right? With an X, right? The male makes Y sperm and X sperm. So that's how it gets decided. So it's not exactly the same in bees, as you'll see in a minute. So, all right. So here's human, just to review. Yeah, there was a really bad picture from the textbook. It was just not very artistically, it didn't have much artistic merit, so I put something good in there. All right. So so the XY system, right? It's the Y chromosome that encodes the male specific. Now, grasshoppers, insects, some insects have a system where the number of X chromosomes determines the sex. Sperm have either one or no X's. And in this case, the male gamete would again determine, because the egg would always have an X. The male could have none or one, so that would determine it. It's a little different. Now, birds are backwards. In humans, the heterogametic sex, the XY, is the male. In birds, the heterogametic sex is the female. The one with the ZW is the female, as you can see in this diagram. So the egg determines the sex. The female determines the sex of her own offspring in a way. It's just a different way. All of these systems are very interesting. Some of the ones in insects have not been well studied, but in bees, yes, very well studied. We know quite a lot about this. Now, bees, if you think about this for a second, I mentioned that I was giving this talk to some of my students, and I said, did you know that male bees in the hive don't have a father? And they're like, what? What are you talking about? What? That's impossible. It's actually true. All right, and believe it or not, that was discovered in the 1840s. So we'll get to that next. This is the haplo-diploid system. This is what bees do. And I'll spend maybe 15 minutes on this. So you'll notice that the female has 32 chromosomes and the male has 16, exactly half. Males come from an unfertilized egg. And you may know that the queen goes out, flies around, picks up a whole bunch of sperm, stores it for years, never flies out again, and uses that sperm to fertilize eggs. But not all of them. The ones that are unfertilized, what do they develop into? Drones, the males, drones, yeah. So the drones literally don't have a father. They come from an egg, right? They do have a grandfather, though. All right, so check out this guy. So this is Jan Zerzan, Polish fellow. and he was a Catholic priest. He is known as the father of modern apiculture, and I had never heard of him before the other night when I was making up, learning new things. He discovered bee space. He figured that out, right? How much space they need to make a removable frame that doesn't get cross-linked with the other frames and so on. And he invented the movable hive in 1830. Langstroth built upon his research. He published a lot of papers, and he was a hero in his nation. He was well known, very, very famous in his day. And he made the discovery that unmated queens produce only male offspring in 1835. Wow. People have been really smart for a long time. You know, it's amazing what people have known about. And so, unfortunately, all this stuff happened right around in his life. Like Nazis came and they tried to claim him as a German because he published in German. Anyway, it's very crazy what happened. You should read up on him on Wikipedia. It's very interesting. Okay, so let's talk about sex determination honeybees. What do you see on the left? You see a lot of holes, right? On the right, you see a nice pattern brood. And we all know we're supposed to look at our brood and see a nice tight brood pattern. But until your talk last time, Mike, I think it was, you mentioned something about double drones and things like that. That actually is what's going on here. So on the left, there's a term for that. It's called a shot brood pattern. And this is what happens in the case when you have a lack of genetic diversity in your queen. I think I can explain, I hope I can explain this clearly so that you can understand why this is happening. All right, so there's a couple of slides that show this. So this is from a paper that I'll show you. I'll show you the title of the paper in a moment. But it gets into how the sex determination works in bees. All right, so well, here's the title. So this is all about a gene called CSD, complementary sex determining gene, CSD. All right. Now it turns out that you need two different alleles of the CSD gene, right, to make a female. Two different alleles, two different versions of that CSD gene, which has a bunch of varieties in the population, like maybe 15 or so different varieties out there in the world. If you have the same one in a drone that's in the female, in the queen, then you can end up with a diploid, a fertilized egg that has one version of the CSD. And guess what? It develops as a male. A diploid male. A male with two copies of all the chromosomes that you would think would develop as a female, but because the CSD gene is the same version, it developed as a male. Does that make sense so far? Who has a question on that? I don't want to go any farther than that. Go ahead.
So the queen knows that something's fertilized, so she'll put the fertilized egg in a small cell, right? But that could come out to be a male.
Let me explain what you're getting at. So the queen, the question has to do with whether your queen is laying an egg to make a drone or a female to make a fertilized egg to make a female. And so yes, the workers make larger cells. The queen will decide not to put sperm on that egg, put the unfertilized egg, and it'll grow into a whole bunch of drones, usually all clustered together at the bottom of the frame. So that's the normal process. What this is a fertilized egg, because she's laying it in the normal middle of the see the picture, right in the normal part of the middle of the brood. But a whole bunch of those, even though they're diploid, they're fertilized, start to develop as males.
Oh, those are drones on the left, the empty holes?
The empty holes were male developing fertilized eggs, which shouldn't happen. Fertilized eggs should develop into females. So the amazing thing is that the bees can tell shortly after that little larva pops out and hatches out of the egg, they can tell it's a diploid drone. Are you okay with the term diploid drone? They can tell. Like, to me, that's a mystery. How can they tell? What is the signal that they can tell, right? And they eat them and get rid of them, and that's why you have all those holes. And actually, it's 50% of the holes. 50%. Exactly 50%. I'll show you in the next figure why it's 50%. Are any other questions? I can clarify the point. So remember, it's the CSD gene that controls female development, but you have to have two different alleles of it to get female development, two different varieties of the CST gene. And this picture shows on the left is your queen, on the right is a male, and you see where it says X, there's an XC and an X. I can barely see this on my screen here, hold on. There's an XC and an XF, and those XC and XF are two different alleles of the CST gene. All right, and on the left side you have unfertilized eggs. Those should be drones, right? Unfertilized eggs, and see how it says male, male, and you have the XC and XF. Those are fine. Those are going to be great little males, right? You see that? And then on the right you have fertilized eggs. And that male has the XC allele, which happens to be one that's also found in the female. So there could be a problem if you have a diploid male that's XC, XC, has the same allele, XC, XC, and by definition then you get a male pattern of development in a diploid fertilized egg. Those are the ones that end up being holes. Those are the ones that end up getting eaten and they don't develop. They're the diploid males. And then you have the normal XC on the right. Those are the ones that become females. Yeah.
But doesn't the queen mate with up to 10 drones? So why is it?
Yeah, if you have enough genetic variation, you would never see this. Would happen if you control the mating so that it is purposely producing 50%. These are purposely made to make this 50%. And then what they did is see how it says mapping population? The ones that turned into the females, they use that to map the location of the gene on the chromosomes. So they look at they were able to home in on where the gene was located by genetic mapping techniques, which are beyond the scope of the talk, but it was important for their study that they had this set up like this. They get a whole bunch of diploid males that don't develop, and then all the females have this XFSC, and they can look and see where there's crossovers between the chromosomes and where they're looking for a place where there's never a recombination event, and that's the locus where they're of interest, that they're zooming in on. Basically, they were able to home in on that locus. Yes?
So, if you're in the lab and you want to make XCXC eggs,
Yep.
and you have complete control over the mating of the queen, pathological as it is, how do you do it?
Yeah, so what they would do is they would So the question is, how would you set up this mating? How would you do this in the lab? You would have to know the genotype of the female, the genotype of the female, which alleles she has, and then you would probably take a drone from that female and figure out, oh, this drone has this allele, and then you would take the sperm from that and a queen from with a known genotype and just instrumentally inseminate the queen so that she only has like one drone's sperm, right? So you do that artificial insemination. It's actually called instrumental insemination. It's another thing I learned, right? So yeah, they would artificially control the breeding, basically. Sort of like Mendel couldn't do that, but they can.
So it's mating with their brothers.
Yeah, which you don't usually want to do, right? And this is what happens, right? It's disaster, 50% death, right? That's not good. So their goal was genetic mapping, not making a lot of honey and having a lot of bees. Exactly. So this is the paper. This is it's from 2003. So it's not brand new knowledge. It's not the latest thing. But the summary of it is that they discover this discovery of the shot brood pattern is very old. But then they used that to map this gene and discover the gene where you have to have heterozygous condition to produce females, two different alleles. If one allele is present, you get a diploid male. Yeah, there it is. So this was in Nature News. You can check this out. There's a link there if you want to go read the news article. And they said that this is a holy grail of bee genetics. It was really something they knew was there, but then they discovered it, right? And more is known about the pathway. At that time, 2003, they just knew this is the gene, right? And now they know what it does. It actually, when that gene is produced with two different alleles, it causes another gene to be produced successfully, splices a gene called feminizer, And then feminizer then goes on and produces double sex, which is a male or female specific version. In other words, it initiates the pathway of sex development. So the practical message for all of us is you get a new queen, check the brood pattern. Right? You've heard that, but now you sort of know why, because there's no guarantee that necessarily, especially in a wild mated queen, that you have enough genetic diversity to avoid this kind of a drone from the same hive mating with the queen, and you might end up with these like a third or a quarter of your brood just missing. So quickly replace that queen if you see that. Now I'm wondering if anyone of us have experience with that in the group. Has anyone had a queen that gave a lot of holes? Maybe you thought, whatever. Have you seen that before? I always see some. Sometimes you see a line of them where the wire is. That's normal, I think. No one? No? There's something to look out for. Yeah.
I got a question.
Yeah.
So it's possible to get a triploid female if one of those diploid males actually mature and do their business.
So that's true. That's why. So the question is, like, if you get if one of those double drones were to grow to adulthood and then produce sperm and mate with a queen, what would happen? You would end up with an inviable offspring, because with 3 sets of chromosomes, it wouldn't be viable.
Oh, so
it wouldn't develop. So this is why they get rid of them. They would not be good fathers, if you will. They would not genetically be able to produce normal offspring. So some plants, you can have triploid, polyploid plants, and they work fine. There actually are multiploid. In other words, this term ploidy means you have whole sets of chromosomes extra. All right, that can happen in some situations in work, but I don't think it works in bees. Yeah.
For the workers, when they go to eat those, is that pheromonally controlled, or do we know?
I have not read what the signal is that alerts them to this situation. I don't know what that would be. I really don't know. Do you know?
Well,
It may be pheromonal, yes, but do they?
it's a level of pheromones shortly after it makes the transition to the larval phase.
Right. Okay, so the signal then may be known for what alerts the worker bees to get rid of the double drones, and that's the levels of a particular hormone or a pheromone rather, that is not a normal amount for a typical worker development. They're like, oh, this worker has too much of whatever, eat that up, I mean, it's amazing, really, that they can do that. All right, so I found a wealth of genetic information at Glen Apiaries. Have you ever visited this site? Has anyone here ever been to this site? They specialize in, have for years specializing in queen development using instrumental insemination. And this is really fantastic. So this is almost the same diagram, if you'll notice, that we had from the paper, which shows a couple of bees on the left-hand side, where the sex allele is shown in blue in the drone on the right, and then red and yellow on the left in the queen. And you'll see that there's no offspring combination where you're going to have like 2 of the same, right? Because you're going to have a red and a blue, or a yellow and a blue in the females, right? Of course, the drones are dictated by the females. See the drones on the left? They're either yellow or they're red, right? The drones, the eggs, each egg still gets a jumble of the genes from the female, but they're all from the female. 1 copy of each chromosome. Which one? It's jumbled up, but so there's diversity, but there's more diversity when you have the 2 parents. Now on the right you have a bad situation, right? The female, the queen, has a red and a blue, and then the drone has a blue. So half of those offspring of half of the females with the red X are the inviable double drones. Excuse me. Yeah? Clear as mud. I hope that makes sense. Now this is a somewhat unnatural situation. Here's a typical situation that was raised a minute ago. What normally happens is if your queen flies out, she's going to go to the drone congregation area. And there's going to be a whole bunch of drones flying around up in the sky with their big eyes, looking for the queen, mate. And so she mates with a whole bunch of like 12 or 15 or 20 or, I mean, it's a crazy sex party. And anyway, she's just getting loaded up with all this sperm. So she has a lot of diversity. And then there's very little chance of, as long as the drones are diverse, of having the double drone problem. Yeah? So that's good. So does this help you understand a little bit about the shot brood pattern and sex determination in bees? It's pretty interesting. You get the idea now. It's a puzzle. Why do the drone males, why they not have a father? Now you can just throw that at people and they'll just scratch their head. They won't understand. Let's talk next about genetic traits of honeybees. And again, a lot of this came from the Glenn Apiaries, Tom Glenn, this figure. So think of some human genetic traits off the top of your head. Eye color, additive trait like that one, an additive trait, right? All different shades of blue. So there's more than one gene leading to eye color, dark brown eye color, right? What's another trait you can think of? Hair color, similar idea. Height, similar idea. Can you think of a single gene, single trait? Attached earlobe. Remember all these things you did maybe in genetics class? Oh yeah, the chin, cleft. There's a forelock, white forelock. There's all these little traits and things that are out there. Now, can you think of any bee traits? Color. Yeah, the color. That's right. There's variations in color, definitely with different like races of bees, like the Carniolans versus the Italians or whatever. Well, let's talk about some ones that are understudy and important to beekeeping. So, I don't know if this one's important. It's really just interesting. So here's one that causes a lack of eye pigment. And it's a recessive gene. So you see this mostly in drones. Because they have only one copy, then you're going to see recessive genes galore being expressed in the drones, right? Because they don't have another copy to cover that faulty gene. So there's an ivory eye color. And I believe this is similar to the white gene, presumably in fruit flies. Maybe it's a similar thing. I think they're blind, which is fine in the hive, right? You don't really need to see, but they're probably not very good at mating, without being able to see. And here's one right next to the other coming out, an ivory eye one. That was interesting, I thought. There's also one called the cordovan body, which I honestly didn't know about until doing this talk. Yeah, and so you've seen these? Have you seen cordovans?
I know.
Yeah. So it's actually been used as a tool by geneticists if they want to tell if their queen is still there or if she's been replaced, because at a glance you can tell, the cordovan queen is there. All right, it's a marker. It causes the black parts of a bee of any kind to just turn this light brownish color. You still have the sort of black-yellow pattern, but instead of black, it's like a brown color. Right? I think it's similar in cats. They have a gene that makes all the colors just turn light, a light color. And so typically this is made in the lab with instrumental insemination. And so over time, if you use a cordovan queen, like she's going to be like a pure, it's recessive, right? So she has to have 2 copies of that gene. So all of her offspring are going to have, all the drones, of course, will have it, right? And, as long as, I suppose, the drones have to be selected, and then, the cordovan drones, so you get 100%, hey, that's on, 100% offspring, all cordovan. And so if she gets replaced or superseded, at a glance, you can just look at the entrance of the hive and start seeing, oh, we're starting to get other colors here, something's wrong, and I have to redo my experiment. So, yeah, it doesn't seem to have a functional effect on the other behaviors and things of the bee. All right, cool. So hygienic behavior is a big one. So this has been known about for some time. Back in the 60s, Rothenbuehler, does anybody know him? Anyway, kind of hypothesized that this was controlled by a couple of genes, a gene for like the uncapping behavior and a gene for the removal of the diseased larva. And it turns out there might be multiple genes like the eye color that lead to this, called a polygenic trait. So that's the thought today. And so if you have enough of these hygienic behavior genes, then you can have a drastic reduction of things like even American foul brood or chalk brood. They'll just detect it and uncap and remove those bad bees, right? And so it can get rid of that problem. I wasn't able to find a primary reference for the actual amount of reduction of chalk brood, for example, but that's one. And so over the years, you would hope that our suppliers of queens, that, from Georgia or California, would be really paying the money to start to get some of these good genetics into the queens that they're giving out, that they're selling, and they're not giving them away. Excuse me. All right, so then the other hygienic behavior is called varroa-sensitive hygienic. So this is like hygienic on steroids. I really don't like that term, but it's funny to use. So it's like extra hygienic. And so in the sense that they don't just detect foul brood or or chalk brood or whatever. They detect varroa and detect that it's in there, that they're fertile varroa, and they remove the fertile reproducing varroa specifically and get them out of there. So the observation that John Harbaugh and Jeffrey Harris made, they worked at the USDA back in the day. Maybe they still do. I believe that's Harbaugh giving a talk. The observation was all of a sudden they saw an increase in the number of infertile varroa mites in their hives. And at first they didn't know what was going on, but then they realized, it's because they're getting rid of the fertile varroas. It's a behavior that's programmed in, and it's genetically encoded. It seems to be an additive trait with multiple alleles that add together to produce this effect. Now, I looked at the original paper and that's cited there in 2009. There's no mention of a decrease in the quality of the bee reproduction, but Glenn mentions on his website that too much of the VSH trait causes a decrease in the amount of brood production. And they were saying that the optimal is about 50% of the alleles, and then you get a lot less varroa and still good production of bees. That's pretty interesting. So a lot is still being figured out. How do they do this? How does this work? What are these genes? And it's very complex. Doing bee genetics is a lot harder than pea genetics, because with peas, you have the female flower, you have the male pollen, and you put it on there, you control it, and you don't have all this storage of sperm from 40 different bees. And it's way harder with bees to do genetics. A lot harder. Hope you appreciate that. So the last couple slides here, a few slides. Think of a bee colony as something you want to breed, right? Because a colony is like a superorganism. You've heard that term. It's like an individual bee is nothing. It works as a unit. All the workers, the drones, the queen, as a unit work together, and that's where the quality is. As the whole thing is good quality. Like, this is a good hive. This is a weak hive, right? Of course, it results from the genetics carried in by the queen, ultimately. But the colony is really a mixture of the queen's genetics plus the sperm from a bunch of drones. It's diverse. Now, when that colony, say it's a good colony, produces a new queen, you're not getting the diversity of the whole colony in that new queen. You're getting half of the good queen, half of the queen, and one drone's sperm. Who knows how good that drone is, right? So it's kind of a crapshoot as to what you're going to get. And propagating a good colony is not as simple as propagating a good, bovine or pig or something. It's much simpler, right? So the future holds the promise of figuring out what all these various genes are that are important for hygienic behavior, for example, and then picking the drones that have those genes and collecting the sperm from those drones, inseminating a queen that also has those good traits. And actually, that could possibly be done in the near future by rapid DNA sequencing. You take a wing clip and send it off and get DNA sequencing done on it. A couple of days later, you get back your genotype information, right? You wouldn't be doing this, but bee breeders would be doing this, greatly speeding up the process of generating better varieties of bees. That's the idea. Yes?
What's that cost?
Cheap. So cheap. I mean, it's going cheaper every year. DNA sequencing, it's done by machines and computers. It's not like you sit there and do a lot of labor, but it's taken years. The question is like, how expensive is it to do those kind of tests? It's well within affordability. Think of the importance of this to the agricultural industry, to have bees not be losing colonies and so on. So I think it's definitely something that can be done. Like previously it wouldn't be doable, and now the cost has come down. So you can get your own human genome sequenced in a couple of days or a week or so for $1,000, something like that. If you're determined to know the right people, it's amazing. Things that took decades in the past are done very, very fast. They're working on sequencers that are the size of a fat USB device, not like the one you have, like the old USB sticks. They're about that size, and they can sequence a human genome in like minutes, literally in minutes. But that's sort of a future technology that could potentially allow us to really home in on the best bee genetics, right? Yes, Mike.
So the really hard part, though, is knowing which DNA you want.
Yeah. So for example, you could sample the sex-determining genes and make sure that they are diverse, just as a simple example, because we know that gene. Now the question is, what are the genes that lead to the hygienic behavior? Like that's the hard part, figuring that out. And so, especially for polygenic traits, it's harder to figure that out. But yes.
So there's no way that you're ready, let's say, attaching a color mark on like cordovan to best quality behavioral trait,
Yeah.
trait B.
What you would do is you would know the genotype of your bee, right, for a whole bunch of certain genes, and you predict that this is going to be a good queen, right? It has these traits, and you might even get a certificate showing that you receive. And maybe they also put the cordovan in there so that you can then tell that, oh, this bee hasn't been replaced or superseded. So it would just be a marker that would indicate that quality. It's sort of like a lab mouse having white coat color, and that's that mouse, right?
No, but you haven't seen anybody using that thing.
Yeah, I don't think they don't do this yet. They don't give you a list of the genes. I think they do the best they can. I was going to show this is an interesting, fascinating image. So kind of hard to see, but at the top you see the collection of sperm from 200 drones. Okay, this is when they do this artificial or instrumental insemination. I was wondering, like, how many drones do they use? Well, they use hundreds of them, so they get a lot of diversity. And they also select the drones. They grow the drones in a special way so they know who these drones are, right? What their traits might be. And then here they are putting in 8 microliters into the female bee anesthetized. And so they actually allow the sperm to sit for a day or so to mix well with itself, so it's all evenly homogeneous. Not homogeneous now, homogeneous. All right, so anyway, so this is amazing stuff that can be done. Just astounds me. I wanted to mention one final closing thing in the last minute or two, and that this came out. There's a New York Times article last week, a news article about this paper, which is astounding. So I was going to spend a lot more time on this, and I realized it was probably ridiculous to do so, because it's a whole separate talk. So it turns out that genes from plants, from plant pollen, when fed to the female bees, inhibit the queen development pathway. So what is it that makes one egg, who's perfectly good, female egg, become a queen versus a worker bee? It's the diet, right? The genes are the same. There's something that's being fed to the bees that makes a difference. Now here's a paper that shows part of that is actually molecules from the pollen that change the developmental pathway of the bee, right? Specifically inhibiting the queen development pathway. And it involves a process called RNA interference, where RNA molecules, which are kind of a nucleic acid similar to DNA, they're in there, they're in the pollen, they go into the bee, and then they shut off genes. They just mess up genes that normally would be expressed, they turn them off. They interfere with the production of certain queen-specific genes. And so if they feed the royal jelly, then they don't have this interference happening. But if you think about it, this is truly amazing. So here we have a gene from a plant that's controlling the genes of a bee, which makes me wonder about my pollen patties. So it's not just that. It's like that, plus other things. But this is a contributing factor that they've just discovered recently and published recently. Yes?
Then that means that, let's say, potentially pesticides or fertilizers or God only knows what, that might potentially affect pollen. If we say we can alter pollen, that would possibly either positively or negatively affect the growth of the bee.
Yeah, well, we know bees need pollen, right? My question that comes to my mind is, do all plants have the same RNAs? Or some pollen? I mean,
Would there be lines, genetic lines in there?
yeah, like, what about the pollen quality? Like, how much of it do they need? Is it just a small amount? Maybe they just need a little touch of some pollen that's everywhere, or a few species of pollen. I really don't, those are the questions that came to my mind. And so, yeah, I think that's the next phase of the study, is to figure out exactly which kind of plants produce these RNAs and identifying them better, because they just chose certain ones. This whole idea of microRNAs, or small interfering RNAs, these little RNA molecules that mess around with gene expression, is all new in the last 20 years or so. Nobel Prize winning work. It's really important stuff. We just had no idea in biology, no idea that RNAs, this RNA molecules, could actually be in controlling gene expression. Right? So it's not just the genes you have that make you who you are, it's which genes are turned on and off. And that can be influenced by the environment, as you see here, by the pollen in the case of bees. There's a whole new level of control, of genetic control going on. So,
Question.
yeah.
So the royal jelly, when it gets processed to make royal jelly, it must get rid of the microRNAs.
I think the royal jelly comes right out of their head. It's exuded from their hypopharyngeal gland, some gland actually. I don't want to say the wrong one. There's a lot of glands, but they produce that. They secrete that out of their bodies, so it doesn't have the pollen per se in it. It's like,
Yeah, I was just saying it gets generated from their body, which obviously the microRNAs didn't make it through the stomach, through all of that, and then through their...
yeah, right, exactly. So it doesn't have the microRNAs. That's the idea. That's the hypothesis.
Right. So it's not the royal jelly that makes the queen, it's the lack of the...
Possibly, yeah. So it could be that the royal jelly just is non-suppressive. It doesn't
Right.
suppress. Yeah.
Yeah, with respect to that pollen and inhibiting, is it possible that the pollen can at some point not inhibit the queen growth, and at some other point it does? So
Yes, there could be a developmental window. The question is what if the pollen's inhibiting queen development? Maybe there's a window during the development pathway when it does inhibit, and later that it doesn't inhibit. No, that's not the question.
What I'm saying at all.
Uh-huh.
I'm saying if the growth of the molecule of pollen that changes its status of development in such that it does not interfere with the growth of the queen, or at a future time it does.
I mean, the pollen itself may be different.
Pollen itself is what I'm talking about, yes.
Yeah, I think, as I understand it, this kind of a thing where you have interference. Well, I think that's more the sex determination. I don't know. As far as I'm aware, the pollen, when it's mature, is out there and they collect it. So plants have mechanisms of, so that's not what you're asking. So I don't know, actually. I mean, I just can't, I can only speculate. But the pollen presumably is very diverse from lots of different kinds of flowers. That's usually considered a good thing. Maybe some have their RNAs, maybe some don't. But I would assume that if the pollen is mature, that it is what it is, which is obviously a gamete for plants. It's the sperm of plants. That's its normal job in the plant world, is to fertilize the ovule down inside the female part of the plant. So in that case, it's just amazing that the bees have a system that uses the pollen to control their own development. Now, did they intelligently decide to do this? No, this is what worked. They use it, and they're stuck with it. So we have to respect the biology and go with it and be aware of it so that we consider pollen quality in the future. Whoops. Something died. Awesome. I think I unplugged something, but that's good. We're good. There it is again. It's back. But that's pretty much it. Unless you have other questions, I'm happy to answer them. Yeah, question.
I have a question. So, should we, as an association, change our protocol for the fair coming up? When a little kid asked me, how do they determine, how do they make a male and a female? And the classic answer was, the female's is fed royal jelly only, and the other one is a combination of both.
Oh, you're thinking of the queen.
Yeah.
The queen development. Yeah, no, I think it's generally true. You could say that, the queen gets the royal jelly, a lot more royal jelly, and the others get the pollen and a little bit of royal jelly, I think. So yeah, that's the key. So it's still true. But now this is the mechanism of whereby the pollen has a big effect on the development. See?
Just get the poison. The queen gets nothing.
The chalice from the palace is a breed that is true. What?
So, let's assume I have five hives in my apiary right now.
Test the one.
Would I be better off what I do when I see some clean cells and it's a real strong hive, you split the hive?
Mm-hmm.
That is to take like brood from a bunch of different hives that I have and put them together to change the DNA of all those bees, mix them all up, because every hive has their own DNA or their own whatever.
Yeah, so
diversify the strain.
So you can put in brood from other colonies into the hive,
Yeah.
but after those newly emerging brood workers do their thing and get old and die, after a while you're just going to have whatever that came from the queen, right? That'll replace those.
Yeah.
So yeah, certainly temporarily it could help strengthen a hive. It's to put good brood into a hive, but it's a temporary thing.
You're suggesting, you want to diversify your hive.
What? Yeah. So why is it weak in the first place? Why is a hive weak in the first place? We're talking about what can we do to make our hives stronger? Is basically what you're asking. So you want to, one thing is temporarily, if it's a weak hive, you can put some good brood from another hive in there. They'll start bringing in food. Maybe they just didn't have enough pollen, and so they'll bring pollen in, and then maybe things will start to pick up. But you got to wonder, maybe there's something wrong with this queen, because that's the ultimate source of that hive's longevity for the future, for the 1 or 2 or 3 years in the future.
So would you suggest not keeping the queen from your hive? In other words, getting a queen from Georgia, has nothing to do with that. Diversify that queen, because if you take a queen from every hive every year, take a queen, you really get the same queen all the time.
Well, you see, yeah, the question is, should I go buy a queen from yonder because it's maybe more diverse? The thing is, if you have a good hive and they make a queen and you allow it to go and mate in the drone congregation, you're likely to get a very diverse queen. And there could be wild bee colonies that have lived for years and decades in your woods nearby with drones flying around. So you could get very good genetics, and they would be locally selected by our local weather conditions by allowing nature to take its course. And to me, that's really interesting. I would go for that. Unless you're in business and you're desperately trying to make honey, where you want to get the best queen and no fooling, make sure that's a good queen, so I'm going to get all the good traits, then you might want to buy a queen. But if you're just doing this as a hobby to learn and let nature take its course, I think is a great way to go. But maybe people have different ideas on that. I'm open to learning, totally open. So
didn't the Varroa start? Haven't they always been somewhere on the planet? They didn't just jump on bees.
yeah, they were imported. What year? Where from? Varroa mites came on into America within the last 30 years or so.
But they were always on bees. It wasn't like they used to be on grasshoppers and they.
No, they're very specific to bees, honeybees.
So we should just get queens
So
from
they-
those that area there, because they're already.
They can coexist with Apis cerana,
There it is.
which is an Asian bee, and have so for millennia.
So they jumped
So
species,
you
or
need
they
a good
jumped.
hive from there, because they probably have kept the alleles around.
Well,
whereas
there's
we're-
probably a good reason why
That's a great suggestion.
whereas
we
can
don't
avoid
use
alleles.
Apis cerana.
Why? Why don't we use Apis cerana?
I don't know,
Oh,
but it's an easy solution, right?
well, it
And they
could be
they
they
coexist,
Yeah.
with Varroa and have so for millennia.
You know, my guess is they don't make very much honey,
Yeah.
or they have tiny colonies. There's something about them, but I don't know. I mean, what do we know?
The reason is that they potentially bring in more diseases than we
Ah.
want to import into the United States.
So we don't have that. It's a non-native to the U.S. bee, and we're afraid to bring in a foreign bee, which could have even more viruses and more problems that would then jump to Apis mellifera, our own honeybees. So viruses jumping from one species to another is exactly what happened with HIV, right? With humans, that came from two different independently, twice from monkeys and chimpanzees in Africa. And when a virus jumps species, often it causes disaster for the other species because we're not co-evolved with it for it to be able to exist at a low level. It's actually not to the benefit of the Varroa generally to kill off and make hives weak because that makes them die, right? So over another 10,000 years from now, guess what? Varroa and Apis, our own bees, will be fine. And maybe it'll take 100,000 years. But eventually, they will come to an agreement and everything will be fine. Now, we're purposely trying to speed that process along using genetic tools. So that's the idea. We can identify the hygienic behavior using tricks, like we purposely, do things, and then we look and we measure, increase the hygienic behavior, anti-Varroa behavior. So rather than let that happen over thousands of years, we try to do it in 100 years, 10, 20 years.
So is that how maybe they're figuring out what the alleles are that are the good habits by comparing it to that honeybee's genetic signature?
That's a good question. Yeah, so whole genome sequencing can help. It's a lot of data. Sifting through it is hard. You still have to probably do the genetic mapping and try to figure out where these genes are located in the genome. There might be ways to take a bee that has a lack of a trait and then a little tiny mini chromosome with random genes on it from another bee, and then put those into your bee and see which of those suddenly gets an improvement. And then you can find which gene it is. Right? That's been done in fruit flies. So there are tools, genetic toolkits that are available in fruit flies that are not yet available in bees, but potentially could be done in bees someday. Are you guys ready for a nap? Oh, this is good. All right.
That's good.
See, I know just enough to be dangerous, but but I really appreciate the opportunity to learn. That's why I got into this hobby. So thank you for listening and being such a great group. Definitely encourage you to check out the Glenn Apiary website because there's a lot more that I didn't talk about in there.